But today's lecture is on discovering Mexico's smallest frogs. And our speaker is Tom Jameson. Tom is a PhD student at the University of Cambridge in the Department of Zoology and the Cambridge University Museum of Zoology. And he also has a lengthy relationship with the Natural History Museum, both from his master's and ongoing right now with our herpetology group at the museum. And Tom's current research is really focusing in on the evolution of large reptiles, particularly things like monitor lizards, and looking at climate change and their ecology and how their distributions are changing through time using a variety of different tools from field work to really advanced modeling. But today he's going to be talking to us about something quite different. He's going to be talking about discovering Mexico's smallest frogs. So with that, I'll hand over to Tom to tell us all about this.
Fantastic. Thank you for that lovely introduction. So yes, as is very appropriate for the weather we've been having today, I'd like to take you on a journey to the tropical forests of Mexico to talk about Mexico's smallest frogs. If we're going to let me change slides. Ooh. Already. There we are. Yes, so as we're probably all already aware, tropical forests teem and buzz with life. But what you might not know is that these forests hold a secret. Down in the damp and dark of the leaf litter, we have a secret world. Down beneath the leaves themselves form a tiny forest that is full of tiny species living lives we know almost nothing about. We've only recently started to discover some of these tiny species living beneath the leaves. And so that's what I'd like to talk to you today about, some of the denizens of these leaf litter, specifically some of the world's smallest frogs, and specifically those from Mexico. So we'll start off by talking about the what, why, and how of tiny frogs before talking in a little bit more detail about Mexico's tiniest frogs. So first off, what is a tiny frog? What do we mean by tiny frogs scientifically? Well, here we're talking about organisms that have undergone the process of miniaturization through the revolution. So miniaturization is a size reduction beyond the threshold at which dramatic changes in morphology, physiology, and ecology occur. So this is where a descendant species is much, much smaller than its ancestors. And in fact, it's so much smaller that its body has physically changed a lot. So its morphology, the way that body works has changed a lot, its physiology, and the way that body interacts with the environment around it has changed, its ecology. And for tiny frogs, we're really talking about the bottom limits of vertebrate life, the world's smallest vertebrates. And so if you look at your thumb, and you look at your thumbnail, if you imagine an organism sitting on that thumbnail, that's about the size of the world's smallest frogs, from 7 to 20 millimeters long, total body length. And so we might then ask, if we know what a tiny frog is, why do frogs get so small? What's driving frogs to get so tiny? The answer to that lies within the leaves. Specifically, there's lots of advantages to living beneath the leaf litter. And so organisms evolved to get very small in order to get beneath those leaves and take advantage of all these wonderful features of this damp, dark world beneath the leaves. And so one advantage to living beneath the leaves as a tiny frog is protection from predators. This land beneath the leaf litter is a complex three-dimensional environment, which means it's a good place to hide from things wanting to eat you. So if you're a small frog, you might be at risk of being eaten by birds, reptiles, larger frogs, small mammals, and so this leaf litter provides lots of different refuges to hide from predators. This leaf litter world is also really nice and damp and humid. So if you've ever gone into a forest and stuck your hands into that leaf litter, you'll realize how damp and humid it is down there. And this is great if you're a frog, because frogs typically have this permeable skin, so at risk of drying out in warm, dry environments. And so living in this nice high humidity environment means they don't have to spend so much time and energy trying to find damp spots to keep wet and moist in. Also, as we've seen already, that beneath the leaves is teeming with various creepy crawlies, with various invertebrates. So if you're a frog that likes to eat this sort of food, then it's really important to get down amongst the leaves so you can access this fantastic food source. So we have lots and lots of advantages to living beneath the leaf litter. And so as a result, we talk about wet forest leaf litter as an adaptive zone for tiny frogs. We have lots of species of small frogs from all over the world, from all sorts of different families that have all independently evolved tiny size to take advantage of this leaf litter habitat. And in fact, the world's smallest frogs, at least the world's 29 smallest frogs, all inhabit this wet forest leaf litter niche. They're all living the same kind of lives. There's a couple of species that don't actually live in wet forest leaf litter, but live in environments that provide very similar kinds of advantages, these dense mossy habitats. So I'd like to introduce you to some species of tiny frog. An important one that you should be aware of is Heterochrony amanuensis, which currently holds the crown of the world's smallest frog at only 7.7 millimeters long total body length. So Heterochrony amanuensis comes from Papua New Guinea, lives in the leaf litter there, and it's not only the world's smallest frog, but one of the world's smallest vertebrates. There's maybe a couple of teeny tiny freshwater fish that just about win out over this frog as the world's smallest vertebrate. This is very much the world's smallest limbed vertebrate, so a truly tiny frog. Another couple of species I'd like to highlight just because they have some wonderful names. We have the fantastically named mini mum from Madagascar. As you can see there, a titchy little frog sitting happily on the thumbnail at only 8.2 millimeters long total body length. And it is equally fantastically named close relative, the fantastic minusculum as well. So we have tiny frogs from all over the world, some of them with wonderful names. So if we know why frogs get so small, we might then ask how. What is going on through evolution? What mechanisms are occurring that is leading to this small body size? So in order to figure that out, we might want to look at those different features that different species of tiny frogs share. And so as we've discussed already, tiny frogs inhabit this wet forest leaf litter niche, but also we can look at other features they share as well. So all species of tiny frog, pretty much all species, are what we call direct developing frogs. So you might be familiar with the classic form of frog development where we have our egg, our frog spawn, that then we have hatching from that egg a larval form, our tadpole, that then metamorphoses into our adult frog. But actually amongst tiny forms, we see this tadpole stage get skipped. We have direct developers. So hatching straight from the egg, we have a tiny version of an adult frog. We also find that our tiny frogs belong to a clade that includes larger direct developing species. So here we have our cladogram, our example cladogram of some of our frogs. So this represents the relationships between them. And here we have our tiny frog and its close relatives. And we can see that this tiny frog here, its closest relatives are also direct developing frogs, but they're larger. So this suggests to us that the ancestor of our tiny frog was probably a larger direct developing species. If we look at species of tiny frogs as well, we also find evidence for reduced and simplified morphology. So what do I mean by that? Well, here we have, as an example, one of my very favorite species of frogs, the delightful pumpkin toadlet from the Atlantic forests of Brazil. And if you look closely at this pumpkin toadlet, you might be able to see that it doesn't quite have the right number of fingers and toes that may be what we would expect from a frog. If you look a little bit closer, we can see that there's only really 2 to 3 very stubby toes, fingers and toes on each hand and foot. And so you might look at this and you might be saying to yourself, it looks like these hands and feet aren't fully developed. So it looks like part of these frogs, the development hasn't fully reached what we would expect for an adult individual. In other words, we have some sort of truncation of development. So looking at all these shared features, there's something special, something strange going on with these frogs. They're direct developing and they have some sort of truncation of development. And this suggests to us that we have the occurrence of heterochrony in the evolutionary history of this group. So heterochrony is the changing in times and or rates of developmental processes underlying the formation of morphological traits. So through the evolution of this group, we're seeing a changing in timing and rate of development affecting the bodies of these organisms. And specifically, we're seeing a change in timing and or rate of development leading to what we call pedimorphosis or neoteny. So this is where the development of traits in a descendant species are truncated relative to its ancestors. Or in other words, we're seeing descendant species that look like juvenile forms of their ancestors. So let's look in a little bit more detail about how this can happen with some help of some useful graphs. So here we have on our x-axis time, the time to which development occurs. We have here alpha, the start of development, and beta, the end of development. Here our y-axis, our shape, our actual level of development. And we have our ancestral species here, an example being our large ancestral frog. Our descendant species here being our small descendant frog. And we can see that this frog can get small through a truncation development by what we call deceleration. So here our descendant species develops at a slower rate than our ancestral species, leading to it to reach a lower stage, an earlier stage of development when that development ends. We might also see a process of hypermorphosis where our descendant species and our ancestral species develop at the same rate, but our descendant species stops developing earlier, leading to this truncation of development. Might also see a process we call post-displacement, where our descendant species and our ancestral species develop at the same rate, and they finish developing at the same time, but our descendant species starts developing later, also leading to a truncation development. So we can have all these different features, all these different changes in timings and or rates of development, leading to a truncation of development in our descendant species. And this leads to pedomorphosis. So a classic example of this that you're probably familiar with is the axolotl. And here we have the retention of juvenile characteristics, the truncation of development, and we can see that very clearly in the external gills of this axolotl. So the retention of these juvenile external gills into an adult individual. But we also see this pedomorphosis, this truncation of development among tiny frogs, where we see the retention of the juvenile characteristic, in this case, the retention of juvenile body size. So now we've got a bit of a background on the what, why, and how of tiny frogs. Let's talk in a little bit more detail about Mexico's tiniest frogs. So first off, I'll introduce you to which frogs we're actually talking about here, how we studied them, what we found, and some of the implications of this work. And so which frogs are we looking at here? Who are Mexico's tiniest frogs? So we were looking at a group which is called the Craugastor mexicanus species series. And so to orientate you, this group of frogs are part of a superfamily of frogs we call the Heterochrony or the Brachycephaloidea, over 1,200 frogs that occur in the Americas. They occur from the south of North America down through Central America and the Caribbean into the Amazon basin and the Andes as well. And here, featured here, we have some examples of the Brachycephaloidea. And amongst this superfamily, we were looking at the family Craugastoridae, which is mainly made up of one genus that we were looking at, over a large genus of over 120 species, the genus Craugastor. So Craugastor means fleshy belly. These are the fleshy bellied frogs. They all share this feature of having these big, fat, fleshy tummies. And these frogs have an awful lot of different common names in different languages, and we can maybe learn some of the shared features of these frogs based on these common names. So one name for this group of frogs, for this genus, are the barking frogs, because some species in this genus, rather than croaking, they make a high-pitched barking sound like a teeny tiny dog. Another name for these frogs are the robber frogs. You can see from these pictures, a lot of these frogs have these dark eye masks. They kind of look like a cartoon robber. And another name for these frogs are the dirt frogs. And this is probably the most illuminating about the lifestyle of these frogs. So people often call these frogs dirt frogs because they're small and brown. So they look like dirt. They live on the ground in the dirt. And where these populations are, they often occur in, like where these species are, they often have really large population sizes. They can be common as dirt as well. So Craugastor frogs occur from the south of Texas and Arizona, down through Central America, some in the Caribbean, just into the north of South America. So amongst this big genus, we were interested in the Craugastor mexicanus species series. So this is a group of 6 Craugastor frogs endemic to Mexico, all occurring in Mexico. There's actually a couple of species we find just in north of Guatemala, but this is a Mexico-centric group. And these 6 species, we have 6 historically recognized species. 4 of these species are relatively large. We have Craugastor mexicanus, Craugastor amiltimanus, Craugastor montanus, and Craugastor sultata, all at around 30 to 40 millimeters long total body length. And then we have 2 much smaller species, Craugastor hobart-smithii and Craugastor pygmaeus, both much smaller at less than 13 millimeters long total body length. So why we were looking at these frogs, what drew our attention to this rather small group of small, inoffensive brown frogs from Mexico? We were looking at this group because there was actually a lot of confusion in their history and a lot of confusion about exactly how many species there were. And so we wanted to try and clear up what was going on amongst the taxonomy of this group, amongst where these species boundaries lie and how we should apply these names. And so we had 6 historically recognized species of frog, mainly recognized in the early 20th century based on different parts of their external anatomy. But when we went into the archives and looked at the original descriptions of these frogs, we actually found that these species boundaries were a little bit unclear. Some characteristics that were meant to define one species we were finding in other species. And so maybe these 6 previously historically recognized species didn't actually reflect the biological reality. Also, many of my colleagues, some of my colleagues working on this project carried out expeditions to Mexico in the early 2000s, looking at various Mexican frogs. And they found lots of representatives of this group of frogs, but they found a lot of specimens that didn't clearly fit the definitions of any of these 6 historically recognized species, suggesting to us that maybe we had new undescribed species of this group. Also, previous genetic work had thrown out confusing results, again, suggesting that maybe there was more than these 6 historically recognized species. And we also have these 2 potentially miniaturized species, these two species much smaller than their close relatives, suggesting to us we maybe have miniaturization going on within this group that hadn't previously been recorded for Craugastor frogs. So we're interested to see if this miniaturization is following the pattern that we usually expect amongst teeny tiny frogs. So lots of open questions, lots of question marks here about what's going on with this group, how many species are there, how did they evolve, all this sort of thing. And so we decided to investigate what was going on. So what did we do? What approach did we take? So we took what's called an integrative taxonomic approach. This means that we investigated various different independent lines of inquiry about where species boundaries lie within this group, and we combined them together to try and figure out which species we had. And so we're doing this looking at museum specimens. And so here we have beautifully illustrated an example of our museum specimen. And we didn't just take one museum specimen, we actually took almost 500 museum specimens. So we contacted museums all over the world, and we had sent to us at the Natural History Museum in London and the Cambridge Museum of Zoology almost every single frog from this Craugastor mexicanus group. So we had hundreds and hundreds of frogs sent to us by airmail from museums all over the world. And with all those frogs present, we were then able to investigate what was going on. So we had various different lines of inquiry. And our first port of call was to look at these frogs' genetics. So from our specimens, we extracted genetic data. And here we were looking at 4 genes, 2 from the mitochondrial genome and 2 from the nuclear genome. And we carried out what's called a molecular phylogenetic analysis. So here we were looking at the relative relatedness of our specimens to see how that related to species boundaries and the genetic distance between our different specimens and our different groups of individuals to try and figure out where those genetic species boundaries lay. We also cracked out our magnifying glasses and measuring tapes and had a look at some of the external anatomy of these frogs. So historically, these species have been defined based on this external anatomy. So we re-examined all our specimens, looking at the various different external features we could use to categorize these specimens. So things like the number of warts on each hand, the size of the fingers, shape of the eye, body pattern, all these sort of features we looked at for all our almost 500 specimens. We also cracked out our measuring tape and measured all the various body axes of our frogs. So this allowed us to reduce each of our specimens to a complicated mathematical equation and carry out what we call 2D geometric morphometric analysis. All this means is we reduce our frog to a mathematical equation, and then we can see how our different frogs group mathematically, how our different specimens group mathematically, to see if that can be used to group our species based on a group of specimens and see how that relates to their species status. We also took a lot of our specimens and we threw them inside a CT scanner. And this gave us beautiful 3D models of our frog skeletons. So as well as examining the external anatomy of our frogs, we're also able to look inside, look at their skeletons and see how that internal anatomy varies and whether that could be used to group specimens to the species level. This also allowed us to carry out a more complicated analysis we call 3D geometric morphometrics. We did this on the skull. So we had our nice 3D models of our frog skulls. We're able to reduce that 3D model to a complicated mathematical equation. Again, that allows us to group our frog skulls based on the similarity of those mathematical equations. This allowed us to see if we can group frogs based on their skull shape and how that relates to species. So this is our integrative taxonomic analysis. We have all our different independent lines of inquiry, and we combine these to see where our species are, where our species boundaries lie. So what did we find? What were our results? Well, our headline result is we discovered 6 species of frog new to science. How wonderfully exciting. And so this is kind of what this phylogeny shows. This is our phylogeny based on molecular data. And so we can see here, we've got all these numbers and letters. So these are our individual specimens we were looking at. This branching pattern is their relatedness. So the more closely branching these specimens, the more closely related they are. And based on the genetic distances, we were able to identify which of these chunks of branches corresponded to a species. And this is our phylogeny based on our 4 genes we looked at. So we combine all the genetic data into an overall picture of relatedness. And so here we found that we have in peach our 6 previously recognized species. So yes, those 6 historically recognized species are all valid species, but we also discovered 6 species new to science. And so in discovering those species, we went ahead and described them, and we went ahead and named them. So here are our 6 species new to science. And so we had Craugastor binatonium here. So bi meaning 2, tone meaning toned. You can see we've got this nice 2-toned coloration. This is the 2-toned frog. We then had Craugastor candelariensis, named after the region Candelaria, where this frog is from. This is also a very appropriate name because when in museum preservative, when in the alcohol and formalin we store our specimens in, these frogs change their texture and their color. So they start to look like they're made out of wax. And in fact, they look like tiny frog-shaped candles, hence the name Candlariensis is very appropriate. We then had Craugastor coyatal. This is probably my favorite name we gave to any of our frogs. Coyatal means frog in the indigenous language Nahuatl, which comes from the Valley of Mexico, where these frogs are from. So this is a reference to human populations that live alongside these wonderful tiny frogs. We then had Craugastor polyclavus, one of our less creative names, if I'm honest. So poly means small, clavus means wart. As you can see from the picture, this is a small warty frog, very appropriate name. We then had Craugastor portilloensis that comes from the tiny little town of Portillo del Reo, so a reference to that little town there in that name. And finally, we have this beautiful photo here. So thank you to my co-author Jeff for this lovely photo of Craugastor rubinus. So rubinus is a reference to rubies because the region this frog's come from is famous for its ruby and garnet mines. So we have 6 species of frogs new to science, and you can see from the measurements here that these are teeny tiny frogs. And in fact, this group of frogs are the smallest in Mexico. Now, who exactly holds the crown of Mexico's smallest frog is somewhat up for debate here. We have several specimens from each species and a fair bit of overlap between our different species. So we could do with a little bit more data to see who averages out at Mexico's smallest frog. But potentially it looks like Craugastor rubinus might currently hold the crown with adult individuals at 10.8 millimeters long total body length. So Mexico's smallest frogs are some truly tiny frogs. But we didn't actually just describe our new species of frog. We're also able to use our analysis to look at some other features of these frogs' evolution as well. And so one thing we found is the evidence for a single miniaturization event in the history of these frogs. So here we have our small frogs in yellow and our large frogs in blue, and we found that our small frogs all are part of a single clade. So all our small frogs are more closely related to each other than they are to any of the large frogs. So this suggests to us that our small frogs have a single common ancestor, and so we probably have a single miniaturization event in the history of these frogs, and then diversification into all these teeny tiny forms. Based on our CT scans as well, we also find evidence for reduced morphology amongst our miniaturized species. And so here we have some of our beautiful 3D images from our CT scanner. And as an example, we have an example of one of our large species of frogs, Craugastor mexicanus, an example one of our small species of frogs, Craugastor rubinus. And here we have, you can see from the skeleton of our large frog, a nice robust skeleton. The skull is nicely fused up and all the bones are sort of where they should be. And if we compare this to our smaller species, a typical example of our small species of frog, you can see there's actually a lot of skull bones missing from the skull of our smaller frogs. And the bones that are there aren't fully fused up as well. Similarly, if we look at the limbs, we can see the ends of the limbs, these limb epiphyses aren't fused up in the way we would expect to see. And this lack of ossification is the skeleton, and the skeleton is something we might associate with a juvenile frog. So juvenile frogs often don't fully ossify their skeletons, but this is in fact an adult frog, and the small frogs we were looking at were adult frogs. We're able to identify that by dissecting our frogs, looking at their reproductive organs, and finding that yes, these are sexually mature adults. So we have a sexually mature adult frog that is retaining a juvenile characteristic of a not fully ossified skeleton. And this might sound familiar because we find that our tiny frogs, our new species of frogs here, share all the features that we would expect to see amongst other miniaturized frogs. They inhabit wet forest leaf litter, they're direct developing, they belong to a clade that includes large direct developing species, and they have this reduced and simplified morphology. Here, reduced and simplified with a lack of ossification of the skeleton. We also find a potential link between miniaturization and elevation within this group. So looking at where all our specimens come from, we find all our small specimens appear to come from the lowlands of Mexico, whereas all our large specimens are coming from right up in the mountains at over 2,000 meters above sea level. So we start to map that out on a topography map of Mexico. Here you can see the different colors. So we have dark colors, high elevations, then these paler colors, low elevations. And we have in blue some of our larger frogs, in yellow some of our smaller frogs. And you can see that these small frogs are typically coming from these lower elevations. And this holds up statistically as well. So here we have 2 species, one large species, Craugastor mexicanus, one small species, Pygmaeus. And these were 2 species we had hundreds and hundreds of specimens for. And so we were able to look at all of those specimens and look at the data associated with them to see what elevation those specimens were collected from. And we found that there's a statistically significant difference between our large species and our small species. Our large species are occurring at statistically higher elevations than our small species. So what could be going on here? Well, we know that there's usually a link between elevation and temperature. So up in the mountains, we expect things to be cooler than it is down in the lowlands. And we know that development can often be affected by temperature. Rates and timings development can be affected by temperature. So here we might have an additional mechanism that is affecting the evolution and development of these frogs, leading to small body size, namely temperature associated with elevation. So what are the wider world implications of this study? I think it's fantastic we discovered 6 new species of frog. That is itself really interesting. But as someone with a conservation background, my first thought in discovering a new species is, what does this mean for their conservation? We can't protect a species until we discover it. And once we discover it, we need to figure out how we can protect it. And so looking at the localities our frogs are from, we can start to draw some ideas about what the conservation status of our frogs are. So here we have maps of Mexico, and mapped out in all these lovely colors and shapes are the localities that our different species of frogs come from. So this is the entire Craugastor mexicanus group, the 6 historically recognized species and our newly recognized species as well. And you might be able to see that some of our species are only known from a single area. So here we have Craugastor rubinus here in red in the state of Jalisco. And you can see that this frog is only known from this single area, from this single hillside and single forest in the west of the state of Jalisco. So this suggests to us that this species is what we call a microendemic. So endemic species are those that only come from a single country, and a microendemic is one that only comes from a single, very, very small area. In this case, this species appears to only occur on a single hillside in this part of Mexico. Now, this leaves this frog very vulnerable to habitat change. If something is to happen to this hillside this frog lives on, then its world habitat is wiped out, and that probably spells the end of that species. And so, as we've mentioned already, Craugastor rubinus is so named because the region it comes from is famous for its ruby and garnet mines. But if these mines are to expand across the state of Jalisco, then we might see the little hillside that this frog comes from get turned into a mine, and that could spell the end of the species. That could be the global extinction of Craugastor rubinus. So it's important that we identify these new species to be able to identify potential risk factors and be able to take appropriate action in response. Another thing we were able to identify from our distribution of species and distribution of our newly described species are potential diversity hotspots. We found a couple of areas in central Guerrero and in South Oaxaca where we have lots of species of the Craugastor mexicanus species series occurring closely together. So we have lots of species in a pretty small area. We have a bit of a biodiversity hotspot. So this suggests to us there's something special about the ecology of this region that's leading to high diversity in this group of frogs. Not only this, but here our frogs act as a bit of an indicator species as well. If there's something special about the ecology of this region leading to high diversity in this group of frogs, it's probably leading to high diversity in other groups of animals as well. And so we can actually use this study to identify previously unidentified diversity hotspots in Mexico. And these might be places that we really want to focus conservation action. And so here we have, again, mapped out in lots of colors and shapes the distribution of our frog species. And we have in green here Mexico's protected areas. And we here again have our biodiversity hotspots for the Craugastor mexicanus species series. And you can see here that there's almost no protected areas in this region. So in Oaxaca, we have almost no protected areas at all. In Central Guerrero, we have a couple of teeny tiny, these are little tiny community run protected areas in this region. So this suggests to us these might be places that we'd want to improve the protection, increase the number of protected areas, and really support those existing protected areas. And so going forward, we'd really like, my colleagues now would really like to be working and are hoping to work in the future with different parts of the Mexican government responsible for protected areas, work with colleagues at Mexican universities, and most importantly, work with the communities that are currently protecting these regions, work with communities living alongside these frogs to strengthen the protection for these tiny frogs to make sure that they have a future beyond us just discovering them. So that's pretty much all from me. But to conclude, what I'd like to take away from this talk is we discovered 6 species of frogs in science. And these frogs are teeny tiny. They're miniaturized, and this miniaturization is an adaptation to living in the wet forest leaf litter, the leaf litter of the forest floor. And this miniaturization occurs through a process of heterochronic pedimorphosis. In other words, through evolution, we have a truncation of development leading to small body size. And in this group of frogs, it appears we might have some link between this small body size and the elevation at which these frogs occur. And many of our newly described species are microendemics. They only occur in a really small area, but this makes them very vulnerable to habitat change. And so conservation action is sorely needed in order to ensure a future for these teeny tiny Mexican frogs. So just before I finish, just like to acknowledge a few people. This wasn't a solo project. I had lots of wonderful co-authors helping me out here. So thank you to the fantastic co-authors, Jason Head, Jeff Stryker, Luigi Manueli, and Eric Smith. As I mentioned, we had specimens sent to us from all over the world, from lots of museums all over the world, but we were particularly supported by these three institutions: the American Museum of Natural History in New York, the Field Museum in Chicago, and the University of Texas at Arlington Museum. So big thank you to them. And a particular special thanks goes to our colleagues and friends in Mexico who have helped at various stages of this project, and the people living alongside these frogs at the moment, and the people who are doing the protection of these frogs in their habitat. So a special thank you to the people of the various states of Mexico these frogs come from. Now, I think that's all from me, and it's over to you for any questions. Thank you very much.
Okay. Well, thank you. And first of all, can I apologize because we've had an evacuation warning going off in the museum, so you'll probably hear it in about 2 seconds, in which case I might hand off to Padma to actually answer the questions. But thank you very much. That was a great talk, really, really interesting. And there are tons of questions from people here. So I will give you the very first one, which is from Professor B.L. Call. What do you think is the evolutionary significance of these tiny frogs?
Oh, that's an interesting question. I mean, what is the evolutionary significance of any species? Yeah, every species throughout its evolution has a unique and interesting evolutionary history. I think for me, with a kind of evolutionary biologist hat on, a really interesting thing about these frogs that I mentioned is their tiny body size and this evolution of miniaturization. And we see evolution of miniaturization, as I mentioned, through lots and lots of different groups of frogs. It occurs independently. And so actually frogs are a really interesting study system for understanding the evolution of changes in body size, the evolution of miniaturization actually across the tree of life, because we have this really large sample size of different species of frogs that independently evolved small body size. So here we actually have another group that are adding another data point to this giant data set of miniaturization that we can use to study some bigger questions in evolution.
Okay, fantastic. Let's see. Another one is, okay, so I think this was in your talk, but maybe just repeating it, from John Howison. Great webinar, Tom. I'm just curious, do all these tiny frog species form a monophyletic clade, or is miniaturism a polyphyletic trait?
Yes, so as I mentioned earlier, within this group of frogs, all the tiny species form a monophyletic clade. So we have a single miniaturization event probably in the past. So all the small species are more closely related to each other than they are to any other species. However, amongst frogs in general, we see miniaturization happens repeatedly and independently. So in that sense, it's polyphyletic. So yeah, so we in this group, it's a single miniaturization event. They form a single clade, but across frogs family tree, it evolves again and again and again, which is really a really interesting thing to study.
Yeah, absolutely. And this next question was definitely something that was on my mind as well. And the question is whether this miniaturization is sort of unidirectional. The specific question is it unidirectional in terms of from larger to smaller, or are there examples of tiny frog species growing bigger again? And I would just add to that 2 additional points. So if you do have things going from smaller to bigger again, do you think that is then potentially paramorphosis? And also, if you looked across the frog tree of life, do you see a trend? Would you see more instances of miniaturization than the opposite?
So I think there is certainly examples of paramorphosis within the evolution of frogs. So we do see frogs where their ancestors appear to have been smaller and they've got larger again. I think as far as I'm aware, there's far fewer examples of that. And it's much more poorly understood. So that's certainly an area where we could do with sort of an extra study of why it's maybe more common to see miniaturization than it is to see frogs getting larger. So an area that's not fully understood, but yes, we do see paramorphosis as well in body size of frogs. I think it's somewhat harder to identify. I think we have lots and lots of examples of small frogs, only a few large frogs, and they don't have so many close relatives. So a lot of large frogs, all their extant relatives are also quite large. So they're a little bit harder to study. But yeah, it's certainly an area that could require more study.
Yeah, I was in the Copenhagen Museum a few weeks ago, and they showed on a tour, and they showed us the largest frog species. And I have to say, it was massive. It was really big one. So let's see. The next question is, are there observations of similar evolutionary processes in toads?
Yes.
Attention, please.
So that's quite an interesting question, the difference between frogs and toads in itself. So all toads are frogs, but not all frogs are toads. So toads are a bit of an interesting name for a group of frogs. So toads has kind of multiple definitions. So we have a group of frogs called the bufonids, so the true toads, that includes all the kind of toad species you might be familiar with. But we also have, so that's kind of an evolutionary group that we call the toads. But we have amongst that group, those bufonids, we have some species that don't look like what you typically associate with toads. They've not got that dry skin and the shorter limbs. Whereas we do see in other groups of frogs, we see the presence of that aren't true toads, aren't bufonids. We also see the presence of toad-like characteristics, so this dry skin and these short limbs. And so toad isn't so much a kind of real evolutionary term or real evolutionary group. Toad's more a way of life. So it's a kind of, you adapt to be a toad. And so, yeah, so all toads are frogs, and we see in various groups of toads, we also see miniaturization as well. So we see examples of, like we saw earlier, the pumpkin toadlet is an example of an organism we might refer to as a toad. Its close relatives have that thicker, drier skin, and it has those short little limbs. And so, yes, we see so yeah, the term toad is a bit evolutionary complicated, but yes, we do see miniaturized toads as well.
Great, thanks, and sorry for the emergency message going off again in the middle of that. So the next few questions are more on the conservation side. So, well, the first one is sort of in between. So, is it possible that some frogs are brand new species evolving in biodiversity hotspots? And I guess an extension of that would be, what do you think is the youngest species of frogs realistically that we are able to identify?
Oh, I mean, that's quite a tricky question. That really depends where we're drawing our species boundaries. So yeah, evolution is an ongoing process, and where one species, where one species ends and another one starts, it is quite a hard one to put one's finger on. But yeah, evolution isn't stopping, and we're certainly seeing there'll be frogs evolving into new species as we speak. But how we actually identify that is a little bit more tricky. Yeah, does that, I think, maybe half answer that question?
Okay, so there are a few here. I'm just trying to group them together. So Max Whitby is asking about the infectious disease currently threatening amphibians worldwide, and have you ever encountered it in your studies? And what is your evaluation of the threat, to some of these new species that you talked about?
Yes, so that's a reference to chytridiomycosis, which is this disease caused by this chytrid fungus. So it's this pathogenic fungus that is affecting amphibians worldwide, actually, and it's causing the largest mass extinction of any single group of animals since the dinosaurs. So this is really a devastating disease that has wiped out literally hundreds of species of amphibians worldwide. And of course, this is a huge problem for these frogs as much as it is for any others. In terms of my evaluation of that threat, yes, an absolutely huge threat, probably the single biggest threat facing frogs alongside habitat loss. Not a huge amount of work has been done on chytrid in Mexico. There's been some fantastic work done by one of our colleagues in Mexico, Oscar, and so that there's been some early surveys of chytrid in frogs, and we've actually found that, yes, it is affecting Craugastor frogs on the west coast of Mexico. So it's affecting this group of frogs, which is a real concern. We've mentioned already, we have some microendemic species. They occur from maybe single hillsides. And so if chytrid reaches these hillsides, if we have the presence of this pathogen reaching these regions, this could very, very rapidly lead to the extinction of these frogs. It's one of the reasons why we actually want to be looking to take this project forward, to get back into the field and to actually see, these specimens that were maybe collected in the early 2000s, do those populations still exist? They could sadly very well already be extinct, and thanks to habitat change and thanks to things like chytrid fungus.
Yeah, it seems like you can hardly discover them fast enough before all these different pressures are present and extinct. There was a question specific actually to the ruby frog. So are conservation efforts of the ruby frog underway?
So as far as that was sort of part of this horizon scanning we've been doing, as far as we're aware, there's no current active conservation in that region really at all. There's a couple of scattered conservation areas throughout the state of Jalisco. None of those overlap with the known range of Craugastor rubinus. So that does leave this, as we said, this microendemic species very, very vulnerable to extinction, especially because that is a region that is sort of being heavily exploited for mining. So that's somewhere where it could really do with some extra protection. But Mexico is a huge country, and there's a huge amount of biodiversity there. So there's a lot of pressures, a lot of sort of competing interests. But which is again why it's great to bring these frogs to your attention. So hopefully we can kind of up their international profile, up their profile, and let's see if we can do something to make sure that they do survive into the future. Yeah,
do you feel like it is hard to actually get people? I mean, people do love frogs, but on the other hand, there's lots of, the classic people are worried about big mammals and less so small things. Do you find it's hard to get attention to the conservation of these frogs?
Well, I think certainly you have almost 7,000 species of frog, and most of them are sort of like the frogs we've talked about today. So they're really small and brown and sort of hard to tell apart. And so it's quite hard to get people excited about them and wanting to protect them. I think something to highlight here is actually how important these frogs are to the ecosystems in which they occur. So these tiny brown frogs, they're living in the leaf litter, and some of these species have these really big populations. So we've got thousands of frogs per sort of every couple of square meters in an area of leaf litter. And this means that we've got these really high population sizes, which means that they're kind of form the base of the food chain, from the base of a food pyramid for these forests. So one of the species we talked about, Craugastor pygmaeus, and actually two of the species, Craugastor pygmaeus and Craugastor mexicanus, that my co-author on the study, Eric Smith, studied based on genetic data, suggests that these guys have these gigantic population sizes, and they might actually be the most abundant vertebrates in an entire ecosystem. And that kind of means they're feeding this entire ecosystem. They're the food source that forms the basis of this whole Mexican forest ecosystem. And so they're ecologically exceedingly important. So even if they might not be the most exciting to look at, if they're small and brown, actually they're what the whole forest ecosystem is based on. And so maybe that's a way we can get across their importance and get people to want to conserve them, because if the frogs go, so does everything else.
Right. I mean, that's an incredible population density. And that's actually a really good answer to the very first question about what is the evolutionary significance of these frogs, because obviously the evolution of the entire ecosystem, they seem really critical. So there's another question.
Do you mind if I ask a question, because we're on the topic?
Sure.
Tom, you mentioned that there are small community conservation areas in some overlapping in the areas where these small frogs are. I was wondering if they played any role in conserving areas where they, where they are, and helping these large populations form, and if frogs can actually be held up as flagship species like tigers or elephants, to conserve certain areas, because as you say, they're holding up a big web. And if that helps as a policy impetus for people who decide where the conservation areas go.
Yeah, so some of the community conservation projects that we kind of looked at that appear to be occurring within the range of these frogs, especially in central Guerrero, these are really tiny little conservation areas. They're kind of on the outskirts of villages, and they're what were often old orchards and vineyards and things that have been turned into these little kind of acre-sized community-run projects. And they're really important because they are some of the only protected areas in these huge regions. And they're mainly just run by the local community. They don't have much support from the federal government or the state government. So it's somewhere where I think it'd be really worthwhile pouring resources to actually support these communities to sort of continue and expand the wonderful conservation work they're already doing. With frogs as a flagship species, I think that'd be fantastic. By protecting these areas, if you're protecting the leaf litter, then you're already protecting these frogs. But if we could increase the profile of these wonderful tiny frogs, and that can be used as kind of a excuse and as a flagship to protect more species, we can use these in the presence of the Craugastor rubinus, that ruby frog in Jalisco, as a reason to establish a new protected area in that state. Then that's not only just protecting these frogs, but it's protecting everything else that lives in that forest in Mexico as well. As I mentioned about those biodiversity hotspots, similarly, if we're creating some new protected areas to protect a hotspot for these frogs, we're probably protecting a biodiversity hotspot for all sorts of other species as well. So yeah, I think it'd be fantastic if we could try and use these frogs as a flagship species for protection in these different parts of Mexico.
So just a few more questions here. And there are lots of comments to thank you for your talk and a lovely story about a tiny frog that was found in this valley, which I invite you to read. So there was a question here that, unless I misunderstood, you said miniaturization arose from elevation, yet all the tiny frogs are found at lower altitudes. Is there a misunderstanding there? Yes.
So we've got, so we're not entirely clear what's going on with the elevation and the body size. What we think is going on is that we have, at these lower elevations, we have different temperatures. And so we'd expect things to be warmer at the lower elevations. And we expect temperature to affect rates of development. And so we're potentially having an effect of temperature on developmental rate, which is then affecting body size in these frogs. So potentially an ancestral frog moves down into this into a warmer area, and its rate of development is affected. So these frogs end up being smaller, and maybe that's an adaptation as well, an adaptation to the different temperature regime at these lower elevations. So we're not fully clear what's going on between elevation and body size in these frogs. Miniaturization hasn't previously been closely linked to elevation. So again, this is something we would like to study in a little bit more detail to figure out exactly what the mechanism is that's going on here.
Okay, and then the last question here, actually, so there's one more after this one, actually just adds another component into your answer, which is asking about hydration and humidity in addition to temperature and elevation, and that the impact of that on tree-derived litter composition, and well, so I'll just read the exact question. As well as temperature, elevation may be affecting the tree-derived litter composition and hydration and humidity. Is there any evidence of that?
Yes, certainly. I mean, we kind of, these frogs occur through a variety of habitats, but they're mainly these sort of mixed forests along these Mexican hillsides. And so, yeah, along that elevational gradient, we would expect different tree communities leading to different leaf-litter communities. Obviously, you get up in the mountains, you expect that kind of sort of more of that mountain fog, so those high humidities versus maybe drier down in the lowlands. And so that humidity effect could certainly be having an effect. That's a great suggestion. Really, we just understand very little about what's going on here in that system. So it's something that yeah, we'd really love to look at more. And so temperature is one thing, but certainly the humidity as well, and what's going on, the difference in the leaf-litter communities would really be worth looking at too.
That's exactly one of the next questions that was just added was, have you linked these species to specific species of trees? But it sounds like that's an open question right now.
Yeah, very much an open question. Yeah, we don't really have the data on that. But yeah, if we were able to look at the system more, that would be the sort of data we're gathering. You know, we can go to each region, like follow an elevational gradient, see which frogs are in each region, and then see how all these different features change, how temperature is different, how elevation is different, how the leaf litter community is different, like the depth of the leaf litter, the different leaves that make up the leaf litter. So there's all these different environmental factors that one would have to look into to get a clear picture of what's going on. So yeah, it would be a great project, wonderful PhD for someone, something to definitely look at in the future.
And it also just reminds me of the fact that, you know, the more we study things, the more we realize we're barely scraping the surface of so much of the natural world. So I think one last question here, which is, can we assume that they manage well living with the loss of part of their hands and other body adaptations?
Yeah, I mean, so yeah, it appears that these frogs get on fine with this reduced morphology, and that's quite an interesting question in itself, is whether this reduction of morphology is just kind of a drift associated with small body size. So whether the adaptation is small body size, and just as a result of small body size, you also get brought along this truncation of features. So you don't ossify your skeleton, you miss some fingers. And that's just a result of small body size through this miniaturization kind of process, or whether actually losing some of these features is also adaptive. So for example, if you're really small, maybe you don't need as fully ossified a skeleton because you've not got as sort of much body mass to support. And so in fact, you don't need to waste resources in having this very sort of resource intensive, highly mineralized skeleton. So maybe actually having a reduced morphology is also adaptive. Again, it's an open question. It's something that we don't really understand of, how much stuff just gets brought along by accident with the evolution of small body size, how much stuff could itself be adaptive. It's a fascinating system, something that people should certainly be studying, something people are studying. And there's so many open questions. And the great thing is frogs are the perfect study system for looking at this because we have hundreds of species that have undergone miniaturization, all in slightly different ways, with different evolutionary histories from different parts of the world. So it's a fantastic, fantastic sample size, fantastic system for studying miniaturization in vertebrate evolution.
Right, because you have several independent instances of that. Great. Well, I think we have put you to the ringer with questions. I honestly can't remember a talk that's had so many questions from the audience. So you've clearly gotten everybody extremely excited about miniature frogs. So thank you very much for an absolutely fantastic talk. And thank you to everybody who attended today.